My name's Rena Michael. For those of you who don't know me, I'm the Executive Director of Weizmann Australia, the office in Australia representing the Weizmann Institute of Science in Rehovot, Israel. Thanks so much for joining us today. We're very grateful to be able to present Professor Roy Ozeri, Vice President of the Weizmann Institute, and Dr. Ron Diskin, one of the many Weizmann Institute scientists who has urgently diverted his attention to the coronavirus. I'll hand over to Stephen Chipkin, the chair of Weizmann Australia, to say a few words and introduce Roy.
You know, these are extraordinary and fast changing times. We're all aware of that. It feels only a few weeks ago, Corona was the name of the brand of a tasty Mexican beer, and Zoom was the sound our grandchildren made as they pushed cars along the floor. And here we are in a new world where Zoom connects us in our isolation. And of course, we're all taking shelter from the coronavirus. And the image of that virus never seems to leave us. It's, we all know it. It's like an intergalactic asteroid with suckers on the end of it, hurtling through space, threatening our world. It fills our TV screens, it inhabits our minds, and we hide and take shelter. But while we're taking shelter, scientists like Ron Diskin and others have a very different relationship from the virus. He's the sort of person who says, come into my lab, pull up a chair or a petri dish. I want to get close to you. I want to know you. I want to understand what's made you as nasty as you are. And while the rest of us take shelter, we live in hope that a scientist somewhere, possibly even at the Weizmann Institute, is going to be able to unravel its mysteries and shut it down before it shuts us down even more so than it has already. Never in our lifetime, I think, has the word scientist carried so much weight, and nor has the Weizmann expression, science for the benefit of humanity, carried such promise, which brings us to the Weizmann Institute and all that's happening on campus there. Roy Ozeri, as Rena has said, is the vice president of the Weizmann Institute, and actually he's a quantum physicist in his own right, but he's also responsible for Weizmann's relationships with the outside world, including Australia. Roy, we're delighted to have you and Ron with us. I pass it over to you to give us, maybe set the scene of coronavirus research on campus.
I think it's a good opportunity to start for thanking you all for joining us, and thank you also for your ongoing friendship and support of the Weizmann Institute. And I think it's special thanks for your friendship during these challenging times, and these are indeed challenging times all over the world and in Israel as well. Passover is probably the most important family gathering in Jewish tradition, and this year in Israel and I guess across the world, it's going to be, the Seder evening is going to be celebrated through Zoom across families, which is really extraordinary. So in Israel, the disease, like in most countries in the world, has been progressing, and the government has imposed restrictions on travel and on work. People are pretty much required to stay at home, and that had an effect within the last 2 weeks. So if you look at the percentage with which the disease, so R0, and maybe Ron would say a few words about it later, but the average number of people that each person who has corona infects is now, I think in Israel, it's down to 1.15 or so, which means that the exponent is slowing down. But still, the restrictions on travel are in place. And that has implications all over. And in particular, at the Weizmann Institute of Science, we're required to work with less than 30% of staff, scientists, students, employees on campus. We had to send about 70% of our technical and administrative staff on leave. And that is, of course, required in order to keep the safety and the health of our employees and contribute to the reduction of spread of coronavirus within the population. That also has impact on our ability as a scientific institute to perform science, perform research. And that stands in stark contradiction with the duty that we have as a scientific institute to really stay at the forefront of the fight against the coronavirus. It's our role as scientists today, as Stephen said, to come up with mechanisms to produce remedies against the coronavirus. And indeed, in campus, over the last 3 weeks, we saw really a spree of initiatives of creativity coming from dozens of scientific groups trying to come up with ideas, steering their research from other domains, whether it's in the life sciences or outside the life sciences. People are now directing their research to try and come with solutions to the coronavirus situation. You'll hear about one such initiative from Ron in a few minutes, but we see initiatives that are coming from the world of drug discovery, that are coming from the world of trying to find vaccines, that are coming from the worlds of trying to improve and expedite the rate with which we are able to perform coronavirus tests, whether it is finding whether you carry the virus, or serology tests, which are trying to find the antibodies in your blood and detect the fact that you have been infected and now that you're already immune. Initiatives that are trying to, through surveys, detect outbreaks of the disease. And there was a nice article in the New York Times a day ago about research that was initiated in the Weizmann Institute. So people coming from the world of AI and computer science. And we as management are trying or are supporting to our full ability this research at the Weizmann Institute. So it's a balancing act, if you like, between the requirements to lower the presence on campus and our desire to support and accelerate research towards mitigating the coronavirus. And to this end, we established a coronavirus response fund. And the purpose of this fund is to financially support the research that we have on campus towards coronavirus research. Some of these studies, because they're trying to find remedies in the short time scale and not within years, so in order to accelerate them, we need the financial resources. That sometimes that's a lot of money. So with that, I would like to turn and present the speaker of today, Ron Diskin. So Ron is one of our most talented young scientists. One did his PhD at the Hebrew University and a postdoc in Caltech, and in 2012 joined the faculty of the Weizmann Institute of Science. Ron's expertise is studying proteins and the way they interact, and especially the way they affect different pathological pathways. And therefore, he's an expert on viruses. And Ron's group had some major discoveries in the last few years about the ability to vaccinate ourselves, to teach the immune system how to fight these viruses through the use of decoy molecules that can serve as antibodies in the immune system. And one group was able to make progress dealing with viruses called arenaviruses that migrate from animals to humans, very similar to the novel coronavirus. So we have high hopes that Ron's group will make similar progress towards the coronavirus. So Ron, please.
Good afternoon, all our friends from Australia. It's a real pleasure to present our own research. So I will actually give you an overview about coronaviruses, and then I will touch a little bit about our own efforts toward developing some countermeasures. I will start with giving you a brief introduction to coronaviruses in general, their history, some epidemiological aspects of the current disease, And I will touch on the virology of the virus, and I relate that to the multiple efforts that are going on today in the world to find ways to stop the spread of the virus, to provide drugs that will be able to treat infected people. And also I will give you our own little contribution toward these efforts, and then there will be time for questions and answers. This is probably a picture that you all seen in the news. This is a micrograph obtained by electron microscopy of the virus, not the actual virus that we are now dealing with, but another virus from this family. And we need to understand that this family hosts many different species. We are actually quite familiar with those viruses. Now they're enveloped, meaning that they have a membrane around them. And on the membrane, there is a special protein that is called the spike protein. And you can see it here. I hope you can see the cursor of my mouse here. So these white densities that surround the viruses are the glycoprotein spike complex, which is a very important protein, and I will get to this protein later on. But this appearance of the spike protein all around the virus gives the name for the virus, because it looks like a corona. This is why those viruses are named coronaviruses. Now, the very first one was actually discovered in the 60s, and it was discovered as a virus that infects chickens, and even nowadays, it's still a problem for agriculture. And there are efforts to combat this particular virus. So you may have heard about some vaccine development that took place in Israel by an institute called Migal. So they actually addressed this particular virus, and now they try to adapt that to the current human virus. But beyond the virus that infect chickens, we also know about viruses that infect humans for quite some time. And here below, you can see example for 2 such viruses. There are more. And those viruses are actually quite widespread. They cause common cold symptoms, sometimes pneumonia. And a large part of the world's population already contracted one of these strains. So coronaviruses are with us for a very long time. Now in the early 2000s, a new virus emerged that was in China, in the Yunnan province. And this virus caused severe acute respiratory syndrome, and that was the source of the name for this virus. It was called SARS. So it emerged in the winter of 2002, and then by the middle of the summer of 2003, it just went away. It was quite lethal. 10% of the people who contracted this virus died. But as you can see, it was self-sustained outbreak. The spread was not severe during the summertime, and it just went away. There was a lot of effort invested in investigating this particular virus, but then since it went away, the funding went down, and and the research also kind of went down over the years. Now, the next encounter with a deadly virus from this family occurred in the early, well, almost exactly 10 years ago. That was the Middle East Respiratory Syndrome, or MERS, as it now called. So it emerged in Saudi Arabia, and the source for this virus were camels. It was discovered that camels transmitted this virus to humans. Now, it emerged 10 years ago, but it's still circulating in the world, so it never went away. And you can see here on this graph how there is a slow accumulation of cases of people that contracted MERS. Now it's very, very lethal virus. 1/3 of the people that contract it actually die, but the spread is very slow. It's almost linear. So the number of people that are infected from each individual that get MERS is on the average of 1. This is why we see this linear growth, and we don't see a large outbreak as we see with the current virus. And of course, the virus that we are now referring to, it's called SARS coronavirus 2. because it's very close genetically to the previous SARS coronavirus. It's also emerged in China, in the Hubei province, and it was reported in December 2019. But the actual first case was in November. And those numbers, I took them, I think, yesterday, and they're rapidly changing. So we are approaching 1 million cases, confirmed cases worldwide. And the death rate is about 5%. And now we can switch a little bit to the epidemiology of the disease. And I want to mention the death rate because it's something that is important to us. Although the average death rate is about 5% currently, when we look at the global numbers, we see huge variations when we look at the individual countries. So for instance, Italy, that is known as one of the most harshly affected countries in the world, they have the highest fatality rate, which is even above 10%, as you can see here. In contrast, Germany, which is very close to Italy, has also a large amount of confirmed cases already, but you can see that the fatality rate over there is much lower, actually slightly below 1%. And I think this huge difference basically tells us about the very important role of the healthcare system. If the healthcare system can provide good treatment to people, then the survival is much better. The fatality rate is much lower, as currently is the situation in Germany. But in Italy, we know that the healthcare system was oversaturated. They didn't manage to provide good care for all the infected individuals. And this is the cause that we see this very high fatality rate. So it's extremely important to have a good functional healthcare system and not overload it with people, because this is the immediate consequence of doing so. Now, other aspects of the epidemiology of the disease. We see a very clear relationship between the age of the people that contract the disease and the fatality rate. And it's very clear that the risk is dramatically increasing with age. And you can see that people that are above 50 and older people have much higher chance to develop a more severe disease that will be fatal eventually. Now, I think we need also to look at I don't know if it's the bright side, but the more relaxing aspects of this disease is that the kids, the young adults, seems to get infected, but they don't develop severe disease and they tend not to die. And I think as a society, we're much more willing to accept this distribution compared to others. And just to put you on some frame of reference, when we talk about influenza, for example, the young kids are actually one of the more susceptible group of people, and we see much more casualties in this group of people. And we should say thank you that at least in this particular case, it's not something that we see. By the way, we don't know what is the mechanism for that. This is still a very important scientific question about this age relationship. Now, people that contract the virus will likely develop symptoms 5 days after the infection. So half of the people will develop symptoms, but you can see this distribution, and some people develop symptoms only after 14 days. This is why the authorities demand isolation of 14 days, taking into account this graph. So by the 14 days post-exposure, if people did not develop any symptoms, they are considered as not infected. Now, Roy mentioned the R0 number. So this is another thing that we need to consider. So the R0 is actually the reproduction number. And you can think about this number as the average of people that will get infected from each individual that has the potential now to spread the virus. And those numbers are a result or derived from mathematical models that are fed with the actual data that is coming from the epidemiology research. So there are mostly estimates and they give boundaries. So if we consider influenza, which is something that we are all familiar with, so seasonal influenza have a reproduction number of between 2 and 3. The H1N1 influenza that was very scary 10 years ago, slightly more than 10 years ago, had a reproduction number of about 1.5. The new coronavirus has a reproduction number of 1.4 to 4. So it's a range. Now, this is the theoretical number. The actual transmission in the population that is called the RE, the effective reproduction number, is taking into account additional parameters like isolation, the very same thing that we are all experiencing now. So a good social isolation of people, social distancing can reduce this number. And if this number gets below 1, and this is the major goal of all the authorities, then the virus is starting to disappear from the population. At 1, it will be a linear growth, and everything above 1 is still exponential growth of the number of people that contract the virus. So let's switch a little bit to the virology. The virus is an RNA virus. Those viruses are viruses that have the capacity to change, and this is something that we need to take into account when we think about countermeasures, because they might be effective against the strain or the virus that is currently causing the disease, but the virus might actually change over time. Here you can see a representation, a graphical representation of the genome of the virus. And each box here is a protein, one protein that the virus is producing in order to help it replicate and disseminate. Now, some of the proteins here are actually good targets for intervention, for drug design, for countermeasures. And I want to mention 3 because they're relevant, and you may have heard about compounds that are relevant to those targets. So the first target is the RNA polymerase. This is a polymerase that basically replicates the genome of the virus, and it's an essential protein. Without that, the virus will not be able to replicate. Also, the virus has 2 different proteases. Those are essential proteins that cut this long piece of polypeptide chain into segments, and it needs to be cut into segments for them to function properly. So if we inhibit those proteases, then the virus cannot propagate again. And the third, well, not to the second, the third target here is the spike complex that I actually mentioned before. This is the protein that decorates the envelope of the virus. And this is a very important target for antibodies and for immunotherapy. So all of those I will mention briefly now. So when we talk about stopping the virus, basically we can think about treatment options that could be effective for treating people that already infected by the virus, that show some disease that need to be taken care of. And we can talk about prevention. So basically going into the healthy population and prevent them from acquiring the virus at the beginning. And then we can also think about some approach that kind of combines those 2 aspects, and it's called prophylactic treatment. So we can take drugs that are good in inhibiting the virus, and we can take and give them to healthy individuals, and that will prevent them from getting infected. And of course, this is not something that you can do for the general population, but you can definitely do that for high-risk groups like healthcare providers. So let's start with off-the-shelf drugs that gives us the most promising treatment options in the short run. So the first target, as I mentioned, is the RNA-dependent RNA polymerase of the virus. And why we consider that as a good target? So here you can see the structure, the molecular structure of such RNA-dependent RNA polymerases from very different RNA viruses. So this is from the SARS coronavirus, from the previous one, not the one that currently infects us, but those proteins are basically identical in those viruses. This one is coming from poliovirus and this one coming from dengue. And just visually, by looking at those proteins, you can see that they're very, very similar. And they're similar because they do the very same simple thing of taking RNA molecule and replicate it. This is why inhibitors that were developed against those proteins for different viruses can cross-react with the current virus. And this is exactly what people all over the world are trying now to explore. And here you can see some drugs that were developed previously, and I want to mention two of them. There is favipiravir here, this molecule that was originally developed to fight influenza. And now there are clinical trials trying to adapt this molecule against the current SARS coronavirus. And the second molecule that I want to mention is this molecule that there is a lot of hope with it. It's called remdesivir. It was developed by Gilead Sciences, and it was originally developed to fight Ebola. But it didn't do such a great job in fighting Ebola, but people saw that it has cross-reactivity with other polymerases of other viruses. So there was an ongoing research trying to find good targets for this particular drug. And it was discovered that it actually inhibits SARS-related polymerases. And this is a study that was published a month and a half ago, but it's basically summarizing results of a very long research that was conducted over several years, in which it was shown that this drug, remdesivir, is effective against MERS. And again, MERS is one of the members of the coronavirus family. So it was an effective as a treatment option for non-human primates that were infected with MERS. And it was also effective as a prophylactic treatment. So to protect non-human primates from acquiring the virus when they were treated with this drug. So we already know that this drug, and it's shown here, is effective against the polymerase of the new virus. And you can see it here with this EC number. So this is the effective concentration that is needed in order to inhibit the virus. And you can see the lower the number, the better the drug is. And you can see that remdesivir is doing a great job. And there is multiple clinical trials going on with this drug. And we expect results within weeks from now. And it might be the first drug that will be approved against the virus. It is given as a compassionate care drug for people in very harsh situation. And it was even given here in Israel to a few patients. But we don't have this drug in any. So it needs to be provided by the company. It's not something that is available in the market, but we have high hopes for this particular drug. Now I want to mention another drug that is shown here, chloroquine, because you can see that it's also very effective against the virus. This particular drug is not working through inhibiting the RNA-dependent RNA polymerase. It has a different mechanism. But this is a drug that is well known. It's chloroquine. It's a drug that was used and still is against malaria. And the big advantage of this drug is that it's freely available. There are many companies that produce this drug. And nowadays, when people are admitted to hospital and they have a very severe disease, they're being treated with chloroquine. So this is the frontline drug that is currently being used to fight the disease. So the pros of these drugs are that they're immediately available for use. However, those drugs were not developed specifically against this virus. So they have fairly low efficacy. Although there is some benefit, they're not as beneficial as drugs that are designed to target this specific virus. So another drug target are the proteases, as I mentioned, and this is a protease of the new coronavirus. And those proteases are very conserved among all the different coronaviruses. And you can see here different drugs that were developed over the years to inhibit the proteases of the first coronaviruses that emerged. And you can see that they are effective against SARS, the first SARS, against MERS. But none of these drugs were ever taken into clinical trials because the motivation was not there. Just to remind you, the first SARS went away by itself. So no one invested funds in taking those drugs and testing them in the clinic. So definitely we can do that now. So those provide very good starting points to develop drugs that will be effective against the current virus. And there is a lot of room for improving those drugs, but that will need to be developed, and that takes time. So now we're not talking about something that will be immediately available as the previous drugs that I showed you. Now we're getting to the spike complex, and you can see here already the molecular structure of the spike complex. This is the protein that decorates the viruses. And this is a very important target because this is the target of antibodies. And antibodies are great reagents, and we have many antibodies that entered the clinic in the last decade or so for various pathological conditions, and they show a lot of promise to fight infectious diseases as well. So there are many antibodies that people have isolated against the previous SARS, and this is an example for one antibody that was found to cross-react with the current virus. The only problem is that it's partially effective against the current virus because the virus is similar but not identical. And here you can see a graph that is showing that sera from animals that were vaccinated with the previous virus have some benefit. They can also neutralize to some extent the current virus. So it tells us that there are definitely antibodies from the previous virus that were isolated against the previous virus that might be effective against the current virus. This is an ongoing effort, and maybe we will find more suitable antibodies. I also want to mention that people that recover now, that were contracting the virus, they're recovering, they have good immune response against this virus. They have antibodies that can neutralize the virus, and potentially they can donate some blood sample, and you can take the antibodies from this blood sample directly and treat infected people. And this is something that this company from Japan, Takeda, is actively doing. They developed a pipeline that allowed them to extract the antibody portion to actually clean it from other components that are less desired, and that makes it a potentially effective drug that will be immediately available, because all you need are patients that are willing to donate blood. And we have now thousands of people worldwide that recovered from the virus, and the number is keep going up. So there will be a large pool of people that will be able to donate samples, and those will be able to serve as a treatment for people that develop a very severe disease. So those are off-the-shelf drugs. I want to talk a little bit about vaccines now. So the obvious target of a vaccine would be the spike complex that I showed you already, that is shown here in this nice picture. And for the previous SARS virus, people have used this particular protein in many different approaches in order to develop putative vaccines. None of them were developed fully and entered clinical trials. But you can see that there are many different approaches that we can use in order to build vaccines. And many of these efforts are also adapted now to the new virus. If I need to make a guess what would be the most beneficial platform, it would be something that looks like that. So this is an example for a vaccine that was approved about 2.5 months ago to fight Ebola. So it's called Ervebo. It is manufactured by Merck. And the idea is very simple. We're taking a virus that is shown here in blue. It's called VSV. That is harmful to humans. And we engineer it in such a way that it displays the glycoprotein spike, the very same protein like this one that Ebola has, to produce it on its surface. And then we produce a virus that is harmful to humans. It replicates, and it displays the protein that is coming from Ebola. So the immune response against this particular virus is targeting these red symbols that are the spike complex of the Ebola, and that confer protection. And actually, this is something that in our lab we have investigated. So we've looked at people that were vaccinated with this particular vaccine, and we isolated antibodies against this spike complex. And here you can see the spike complex of Ebola. Now this is the spike of Ebola, and you can see the antibodies here decorating the spike. And from that, we can learn how the antibodies target the spike and what is the immune response that this particular vaccine is eliciting. And the bottom line from our research is that we have a very good immune presentation of the desired immunogen using this platform. So I think this would be a very good platform, and already people have started to produce that to develop vaccines against the new SARS virus. Now, when we talk about vaccines, we need to overcome a few bottlenecks. So the first one is manufacturing, and companies have started that already. Now, the next step is to show that these vaccines are actually useful. And for that, we typically use animal models, and we still don't have good animal models for the current virus. So this is a major bottleneck. It could be simple animal models as mice. It could be more complicated. So we still don't know how to test those vaccines. And again, once it's shown in animal models, then it needs to go into clinical trials, and those can take a lot of time. So by far, vaccines is the way to stop the spread of the virus. This is clear to everybody. However, that will take time. Okay, it will take a lot of time until we'll have a vaccine in our hand that is approved for usage. So I want to touch on also about specific drugs or developing specific countermeasures against this virus, and with that I will end my presentation. So I mentioned antibodies, and these are a few examples from the literature about the use of antibodies or monoclonal antibodies that we have isolated, characterized, and we know how to manufacture in order to target infectious viruses. So we can see examples for Ebola, for HIV, and there are other such pathogens that we know we can treat with monoclonal antibodies. So this is very, very promising approach to isolate antibodies that will target this virus. Now, it's promising because we know that antibodies are safe. We can administer antibodies to people without causing any harsh side effects, and they can be very or extremely potent in inhibiting the virus and recruiting the immune system to fight infected cells. So there is a large effort now in the world to isolate such monoclonal antibodies. We also engage in such efforts, both in collaboration with other groups in the world and in a very local effort at the Weizmann with the lab of Ziv Schulman. And we're making progress with that, and we should have antibodies in our hands in the coming few weeks. And of course, we will need to characterize those antibodies, and it will take some time, but this is a very promising lead in order to fight the virus. Now, antibodies are a great immunotherapeutic approach, but there are other ways of doing that, and this is something that we are doing in our lab. And we are developing something that is called immune adhesions. Now, immune adhesions are antibody-like molecules, and you can see a schematic diagram of such a molecule here. So it's very similar to what a typical antibody will look like. There is an active domain that can bind the virus, and then there is the Fc portion of the antibody. This is the leg of the antibody. You can think about that as a flag that can mark infected cells to the immune system, or to recruit effector functions of the immune system. So this is a very important aspect of the antibodies. So we developed those immune adhesions to target viruses, and we did that to a different group of viruses that we study in the lab. They're called arenaviruses. There are also viruses that transmit from animals to humans. And we showed that we can do that, and that it's actually a very effective reagent. If we think about the current virus, this would be the active domain that we will use. So this is a structure that was published only a few weeks ago, a couple of weeks ago. And here you can see very, very nicely, this is the human protein that is shown here in pink, sitting on the membrane of our cells. And this is the viral protein that is targeting this receptor. So we have very specific binding between these two proteins, and we can take advantage of that. So we can engineer this protein, we can cut it here and use it as the active domain, as the decoy molecule that will attach the viruses through this particular interaction. So we kind of bypass the need to isolate antibodies, because it takes time, and that could be achieved in faster timescales. And this is exactly what we do now. Now, the big difference that we introduced to this methodology is the idea that viruses that come from animals and infect humans are actually adapted to their animal hosts. It's a coincidence that they can also use the human receptor, but they are not fully adapted to this receptor. And that gives a very interesting opportunity, because if we now take this particular protein, but we take it not from humans, we take it from the natural reservoir, then the binding to the viral protein would be much tighter. And the efficacy of such a reagent will be much higher, and it will be more relevant for clinical use. So this is what we're trying to do. Here we have the complexity that we don't know exactly what is the animal host that delivered this virus to humans. So we are doing some screening, and we're using computational methods in order to find the right targets. But this is a work ongoing, and we are making progress here as well. So with that, I will end, and we have some time for questions and answers if you have, and I'll be able to take them.
Thank you very much for taking the time to prepare that presentation at fairly short notice. I have a couple of questions. One question is, do you expect the novel coronavirus to mutate? I guess you did sort of say it already has mutated. How fast does it mutate, and what are the thoughts on that?
As all RNA viruses, we certainly expect this virus to mutate. There is already a repertoire of isolates that people have sequenced from all over the world. First of all, it provides a very, very good tool to track the spread of the virus, because it provides lineages that we can follow, and we know how viruses got to different places in the world just by looking at the sequences and finding the small changes that occur in their genome. Having said that, we are not expecting to see very rapid changes that will render it completely resistant to therapeutics, as we typically see with HIV, for example. It's a known virus that mutates very, very fast. Also, there is an interesting point. This is a single-stranded RNA genome, meaning that there is one piece of the genome of the virus. This is very different compared to influenza, for example, that has several pieces of its genome. Now, influenza can reshuffle the genome all the time. It can take different pieces from different strains and have a mixing. This is why each year we have a new influenza strain, different age, different N, and this is because of this mixing. we are not expecting to see that with this current virus because it has only one genome. So it will mutate, yes. I don't think it will be such a problem like HIV, and we're certainly not going to see something that looks like influenza, in which we'll have a completely new strain each year.
A few more questions. One is IDEX, a veterinary diagnostics company, has published a paper indicating that 3,500 randomly submitted samples for respiratory and fecal found no evidence of COVID-19 in the companion animal population. Do you believe companion animals could become a vector?
First of all, I just want to mention that the data is accumulating extremely rapidly. There is a flood of literature coming, and it's hard to follow. This morning, I got an indication that cats were shown to be able to carry the virus. However, it doesn't tell us necessarily that cats can get sick and that can spread the virus. So it's a very different thing. A virus can infect an animal or a host or even humans and not be propagated further because the viral loads would be maintained at the lower levels. So merely the fact that animals might be infected by the virus doesn't necessarily tell us that they will be able to spread the virus further. But we should keep an eye on that, and this is something that might happen because they are mammals. If we're talking about cats, dogs, yes, certainly.
Another question, who is likely to carry the most effective antibodies? I imagine that's relating to people who have survived the infection and maybe according to demographics, young or older.
The assumption is that each infected individual who recovered from the virus have developed good immune response. True, there will be variations, and this is why approaches that rely on taking antibody samples from recovered patients and administering them to people suffer from this non-consistent efficacy, because some samples will be more effective, some samples will be less effective. But overall, each people that recover should have reasonable antibody response against the virus.
How long till a vaccine might be available? For example, the Miguel proposition, and compare that to the timeline for the antibody development that you also suggested, the decoy system.
I don't know the exact numbers, but there are multiple vaccines that were already manufactured. So for companies, for the big pharmaceutical companies, it's quite easy to take their already developed platforms and adapt them to the new virus, like the one that I showed that came from Merck. So the vaccines are there. Now, the big issue is to demonstrate that they have the capacity to protect people and that they're safe. Now, assuming that showing safety is something that could be done on a relatively short timescale, and some phase 1 clinical trials have started already, that could be shown in a matter of weeks, maybe months. But to demonstrate the ability of such vaccines to protect, that will take a long time. I can't tell you exactly how much time. Maybe there will be some regulatory exemptions that will be applied in this case. But this is a very important thing, because you don't want to administer something to the population and then discover later on that it didn't really protect. That will be actually quite devastating, because people will walk around thinking that they are protected while they are not. So this is a major bottleneck, and that will take a lot of time, probably a year from now, at least.
And in terms of the sort of decoy strategy, the antibody.
So the antibodies, we expect drugs, specific drugs, not off-the-shelf drugs that have mild efficacy. We expect specific drugs to be ready in much shorter timescales. Antibodies, for example, are already isolated, such antibodies might be available in a matter of 2-3 months from now.
Given the differing intensity of the contagion in Wuhan, New York, Northern Italy, what impact do you believe does air pollution have on susceptibility to the virus?
I think it relates to the risk factors in general. We know that some conditions are associated with higher risk, like medical conditions, but definitely environmental conditions will affect. So just think about people that smoke. I think they belong to higher risk population in general. So air pollution in general, whether it's coming from smoking or from the environment, might be a risk factor.
Regarding the lockdown of population isolation strategies, do you think that after sort of quite a few long months of most of the population in lockdown, we might then all come out of lockdown and then find ourselves in a second phase, a second round?
That's an excellent question. So I think the answer is definitely yes. The virus is here to stay. It's the global spread of the virus at this point tells us that we won't be able to eradicate it. So it will keep on emerging time after time, but for the next round we'll be better prepared. So there is a rush now to strengthen the healthcare system, to buy more respirators, and we'll have better drugs, and we'll have better means to fight the virus. So it will emerge again and again, but we'll be more prepared to it. And slowly the population will acquire immunization, natural immunization, because we'll have more and more people that already recovered from the virus. And as far as we know today, people are not likely to be reinfected once they recovered, at least not in the short term. How long this immunity lasts, we still don't know. That's a good question, and I guess we'll discover that in the future.
What's your opinion on the cooperation between global research entities, and is China freely sharing its knowledge?
Well, the simple answer is that I don't know. I'm reading the news exactly like all others. I think there is, at least in the scientific world, there is a lot of cooperation. People are sharing ideas, reagents. One of the ways to disseminate knowledge very, very fast is to deposit papers in repositories prior of their official publication. And for us scientists, this is a very important tool to disseminate the knowledge extremely fast. And we see, so people can decide either to do that or not. And we see that a lot of people now decide to do that. So meaning that they're sharing their initial observations very, very fast with the community in order to help advance the research faster. And I think this is a very good indication for the willingness of people all over the world to really collaborate in a way that will eventually benefit us all.
One final question, because we're reaching 7 p.m. here. And I'm sorry if I didn't ask all the questions that were asked, but that were written down, but I've tried. What do you believe we have to be optimistic about? Where are the areas of hope?
We know that all the upper respiratory tract disease or viruses are slightly attenuated during summertime. It's not that they're going away completely, but they spread less efficiently during summertime. And I think we can hope, and there are some even indications that this is indeed the case. So it's not that the virus will go away, but it will allow the governments to have more time. And I think now in Australia, the spread is not so severe as in the States, for example. And I don't know what are the countermeasures that the government is now preparing, but they should prepare very strongly to the virus that will peak during wintertime.
Thank you so much, Ron, for your insights and for your very hard work on this. I think the world's very grateful to all the scientists at the Weizmann Institute and around the world for the work that they're doing. And thanks, Roy, also for steering the ship and doing such a great job together with the rest of the management team. And thanks to Heidi and Kelly for organizing this live webinar. I'm sure everyone has learned a lot, and we'll have, if we could sit around a dinner table with people and share our knowledge, we would, but we will certainly go away with a lot to think about, a lot more to think about. So thanks and good luck, and stay well, everyone, and stay safe. We'll hope to see you again soon.